laboratory-based scientist, and for the last 25 years of my career, I've been trying to make a vaccine to HIV. And now we have 3 vaccines for COVID in less than a year. How did this happen? Well, the COVID vaccines benefited a lot from the years of investment in HIV vaccines. And what I want to tell you about today is how we can use that momentum to speed up the development of an HIV vaccine. But first, let's look at these 2 viruses. They certainly have some similarities. As you can see, they are small microscopic viruses, and you need very powerful microscopes to see them, but there's also some significant differences. So SARS-CoV-2, which is the virus that causes COVID-19, is spread via the respiratory route. So that's simply breathing, and it means that everybody is susceptible. This virus causes an acute infection. That means it's very short-lived, and the body is able to clear the virus. And as we know, that happens in most people. And that's because our bodies make an immune response to the virus, particularly neutralizing antibodies, which develop in almost everybody. Another important feature is that this virus is very stable. So the 60 million people around the world that have been infected with this virus are infected with a very related virus. There are some differences, but it is very similar. Now, HIV, in contrast, causes a chronic infection. And that means it's a lifelong infection. And that's because this virus integrates into our genetic material, into our DNA. The other major factor is that the neutralizing antibody response here is very slow and ineffective. And then another major hurdle for HIV vaccine development is the virus mutates, and it really is a moving target. Now, vaccines have been one of our most successful ways of dealing with infectious diseases, and most vaccines work by inducing neutralizing antibodies. These are part of our immune response. And neutralizing antibodies work because they specifically bind to parts of the virus. In this case, it's the spike protein which surrounds the SARS-CoV-2 particle. So it's actually a physical interaction. The antibody binds to the spike and stops the virus from infecting cells. And that's what it means by neutralizing. So let's have a closer look at this SARS-CoV-2 protein. So this is what causes the spikes or the corona around the virus. And scientists have been able to look at this at what's called a very high resolution. And in fact, again, it's HIV scientists who turned their attention to looking at the SARS-CoV-2 protein. Partly that's because of the urgency, but also because a lot of funding was made available for people to study COVID-19. But what we've learned from the structure of the spike protein is that the part that's targeted by the antibodies is a very focused part. That's the part that's colored in red. And so that is the major target that we're looking, because that is the part of the protein that binds to the cells. Now, a number of different vaccines have been made against COVID-19. And again, it's really benefited from the vaccine development that's been taking place for decades with HIV. And so this just shows you some of the different approaches that have been taken. And many of you will now, I think they're now household names, some of these vaccine manufacturers. So the genetic vaccines, which are composed of the RNA or the DNA of the virus, of the spike protein, are used to stimulate an immune response. These same genes are also put into what is called a viral vector, so it's a harmless virus, to actually carry these genes into the body, almost like a Trojan horse. So in these kinds of vaccines, the body is in fact making the spike protein. We're actually making our own vaccine in a way. The third kind of vaccine is called a protein vaccine. And this is where we make the protein in the laboratory and then use that to immunize people. And this is a much more traditional approach to vaccine development. And it takes a lot longer and is actually much more expensive. So there's certainly, and it's been widely reported in the media about, this incredible effort to develop a COVID vaccine. And now we know we already have 3 that have shown that they work, that they have a high level of efficacy. The 2 RNA vaccines were shown to have over 90% efficacy, and the vectored vaccine also had very good efficacy. But as you can see from what's called this pipeline, there are another 10 that are still undergoing testing, and certainly are, we suspect that they're also going to work. And you can see that in the earlier phases of testing, there are a large number of vaccines. So we have a very, very robust pipeline for COVID vaccine development. And although none have been licensed as yet, that is also very, very imminent. In contrast, in the 20 years of doing HIV vaccine trials, we've only ever done 6 efficacy trials in 20 years. So just to give you a sense of the difference in how we're tackling these 2 viruses. But I don't want to make it sound as if HIV vaccine development is easy, because it isn't. It's actually one of the biggest challenges of our time. And that's really because this virus does change. So we, the laboratory that I run at the NICD, has been looking at how to develop these broadly neutralizing antibodies in infected people. And we discovered during this process that the virus is changing to create the kinds of stimuli that you need to get the right kinds of antibodies. And so this evolutionary process is now being incorporated into vaccine design. And so the paradigm is that we'll need to have different vaccines in order to shepherd the antibody response along so it can become a neutralizing antibody response. And that's just shown graphically here. So this is the equivalent of the spike protein in HIV is called the envelope protein. And so each of those different colors shows that it's a slightly different envelope protein in order to basically, as I say, coax the immune response along the right pathway to make neutralizing antibodies. So certainly in terms of vaccine development, HIV presents some significant challenges. So I participated in a COVID vaccine trial, the Novavax trial, that's been run by Wits University. And I did that because I really believe in vaccines. I believe in the power of vaccines. And also because I am vulnerable, just like everybody else, to being infected by SARS-CoV-2. So I had 2 vaccinations, and I sort of imagined this protein circulating in my body and my B cells trying to hunt it down so it could produce the right kinds of antibodies in order to protect me. During this time, one keeps a vaccine diary. You keep track of your temperatures, whether there's any swelling or redness. And there really was very minor, pretty similar to an insect bite, that really resolved within a few days. Of course, I don't know yet if I'm on the placebo or the active arm of the vaccine. That will only be found out later. And that's really a critical thing for determining how a vaccine works, is to measure the immune responses in people who've been vaccinated and to determine what's called the correlate of protection. So we need to know what levels of antibodies the vaccine needs to stimulate in order to protect people from infection. And that's what these trials are going to show. So a very critical thing. And in fact, once we have the correlate of protection, actually the vaccine development process is going to speed up even further. But I also, we've been learning as we go with this epidemic that we still have many unknowns. So we don't know how long immunity is going to last. We don't know if these vaccines are going to work for everybody. Though there's certainly good signs that they do work in older people, who of course are a major target group. We're not sure if we're measuring exactly the right immune responses, because there may be other things that we are still to discover. And I think a very critical thing is, will the virus change in the same way that it changes with HIV and represent a problem? Once a number of people have immunity to SARS, it's going to put pressure on the virus. And so that is one of the things that we certainly need to keep track of. So now I want to look at the global devastation caused by these two epidemics. So the top graph shows the number of deaths caused by SARS-CoV-2 per week since the start of the outbreak. And what you can see is in the purple is the first wave that occurred in Europe, and you can see the resurgence now that's happening. And then you can see the high level and ongoing level of deaths in the Americas. And what is most disturbing about this graph is that the resurgence levels are higher than the initial levels, and really speaks to the urgent need to start deploying these efficacious vaccines. Because if we don't, these waves will keep happening. This virus will become endemic, and it will keep spreading amongst susceptible people. So far, in 11 months, 1.4 million people have died of COVID-19, and we certainly don't want to be getting to the levels that we see with HIV of 35 million people. So the reason why there's 35 million people who have died from HIV is because HIV has been with us for a very long time. And as you know, we don't have a vaccine. But what we do have is we do have good antiretroviral therapies. And you can see the effect of those on the graph below. So at the peak of the AIDS epidemic, approximately 1.6 million people were dying per year. And that decline was when antiretroviral therapy started being widely used. But it's not going to get us to zero, because antiretroviral therapy treats infected people. What we need to prevent people from getting infected, and that's why we need a vaccine. So we need that vaccine to get that curve right down. Just last year, 2019, over 700,000 people died of HIV. So the other important thing about this graph is the colors. So you can see that it's mostly red. And that's because HIV is mostly affects the African continent. But we also know historically that vaccine development, Africa's been very neglected in terms of developing vaccines for diseases that affect the African continent. So what can we learn from COVID-19 that will help us to develop an HIV vaccine more rapidly? So what we need to be doing is developing and testing multiple candidates. We need a robust pipeline, the same way that we're doing with COVID. We need to be testing lots of different concepts, testing them all at the same time. We certainly need to be testing the mRNA vaccines, which were originally developed for HIV, but now are being tested for COVID. We need to go back, and we need to be doing that. We also need to run parallel processes. So while we're testing, we need to be manufacturing, and we need to be seeking approvals. It's high risk, but it fast tracks the process. And we certainly need more money. So COVID-19, the amount of money spent on COVID-19 is 8 to 10 times higher than the funding for HIV vaccines in any given year. And we know that money drives innovation. It attracts the best scientists and the best industries into this area. It helps us run more trials. Vaccine development is expensive, and it is high risk. We also need more industry involvement. We've certainly seen that in COVID. Many industries, including industries that have never been involved in vaccine development, are now getting involved. But we do need to incentivize them. And one of the ways to incentivize them is, of course, to give them advance commitments. So our governments need to do that. They need to say that we will buy the vaccine if it's shown to be safe and efficacious. There's no doubt that the COVID vaccine is going to have a major impact. It's going to turn this epidemic around. It'll save lives, and it'll save livelihoods. What we need now is to also develop a vaccine against HIV so that we can do the same. We need to attract the best people into this area. We need to use what we've learned in developing COVID vaccines. We need to apply that to HIV so that we can have an AIDS-free world. Thank you.